All right, so we're going to talk first about comparing the two processes of mitosis and meiosis. I'd like to do that first because that's a little bit of review and then a little bit of going forward at the same time, which is always a good thing. So let's just make a T-chart here. Sorry, I'm trying to draw straight lines as much as possible. We'll put mitosis that we did in the last unit on this side, and we'll put meiosis on this side. Okay. So in terms of purpose of each, in terms of the purpose of each of these processes, right? Purpose for both. Let's start with that. All right. Purpose of mitosis. There's actually three. In a multicellular organism like a human, why would any of our cells be doing mitosis? Three reasons. One, organism to grow, right? One is for growth. Another reason why cells would divide in an adult body is if you had damage and you're repairing it, so repair, right? That's another reason. And the third reason, which is different from repair, would be replacing cells that have gotten worn out. So growth, repair, and replace. Those are the three reasons why mitosis or cell division, that is mitosis in nature, would happen in a multicellular organism like a human. Remember that you don't get you don't grow because your cells are getting bigger. You grow because your cells become more numerous. Okay. So now, conversely, go over to meiosis now. What's the purpose of meiosis? Maybe a little bit easier. If you are thinking reproduction, that's correct, but we need to be a little bit more specific than that. Meiosis is, purpose of meiosis is to make gametes. For use in sexual reproduction. There, purpose. What's a gamete? Give me two examples of gametes. Sperm and egg. Very good. Sperm and egg. You guys got it. Sperm and egg cells. Right. Sperm and egg. Okay. These are used in sexual reproduction. All right. That's the purpose. Now let's go down a little bit more and add to this. Okay. Now we've done the purpose. Now where? What's the location? Location. What kinds of cells do these two types of cell division? Right. Okay, you're piping it in. Good for you. Location of mitosis is somatic cells, right? Somatic cells of the body. Right, so body cells. And there are, you remember, there are some body cells that are going to divide a lot, and some that are capable of mitosis, but often don't divide. They go into that G0 phase, right? So you sort of have to take that into account. But all of the trillion of your body cells that are not germ cells in the ovaries and testes, right, are going to be able to do mitosis, okay? The location of meiosis is more specific. If you said ovaries and testes, you are correct. Ovaries and testes are the organs, more specifically in cells that are called germ cells. Germ cells in the ovaries and testes. These are special cells whose fate is to go to meiosis. Not every cell in the ovary and every cell in the testes is going to become a sperm or an egg. There are other supportive cells that make up those organs that are not going to be doing meiosis. It's only the specific germ layer of cells that are actually going to undergo meiosis. Okay. Okay, so we've got purpose, we've got location of each. Now let's go more into the mechanics, just overall mechanics of the two, before I get really more specific. So in terms of haploid and diploid, in terms of what's going on with the division, and I'm going to be very general here, I'm going to get more specific later, but right now, just general mechanics, generalized process, right? Oops. With mitosis, you're beginning with a parent cell that is diploid, right? 2N, diploid. 2N stands for diploid. Over here, the general process, same thing. You are still beginning with a 2N diploid parent cell. In terms of how it begins, they're both the same. After that, it gets a little different. Mitosis goes through anaphase and telophase, and metaphase, anaphase, telophase, and goes through one division, which gives you 2 daughter cells. That are still diploid and are genetically identical. Genetically identical. If you are trying to replace cells that have been damaged, like red blood cells or white blood cells that get bumped around in your bloodstream and need to be replaced, you don't want to be messing around with their genes. You want the new cells, the new daughter cells that you're making in the bone marrow to replace those blood cells to be exactly identically the same as the ones that you lost, because you're trying to maintain your body. You're not trying to evolve. We don't evolve as individuals. We'll talk about that when we get to evolution. So mitosis keeps everything the same because you're growing, you're repairing, you're replacing. You want to keep everything genetically the way all other cells are. Otherwise that would be very chaotic. Okay, meiosis, on the other hand, different story. With meiosis, you're going to go through 2 divisions instead of just 1. Right? So you went through 1 division here. Single division with mitosis, right? Prophase, metaphase, anaphase, telophase, right? With meiosis, you're going to go through 2 divisions, right? The first division, meiosis 1, results in the first separation of the homologs. So you go from diploid to haploid in the first division. And then you're not finished. Then you separate the last sister chromatids in a second division. It's supposed to say 1N, not in. Okay. So you wind up with 4 daughter cells instead of 2. All are haploid. There's a second here. They're all haploid, and they are genetically different from each other. Okay, so there are some differences between these 2 processes. Mitosis, Single division does not shuffle any of the genes. You're still creating more diploid cells that are going to replace or repair or allow an organism to grow. Very important process, and can happen in all of the somatic body cells. With meiosis, this only happens in the germ cells of the gonads, testes and ovaries in humans. They're going to go through 2 divisions. There's going to be a tremendous amount of gene shuffling, where the chromosomes are going to shuffle the genes around, and we'll see that in detail in a minute. And you are creating 4 daughter cells that are going to all be haploid. They're only going to have half the genetic information that you need to make a human being. They're all going to be genetically different from each other. So the parent cell is creating 4 genetically unique daughter cells that will then be used in the reproductive process and unite during fertilization to create a new individual that is genetically different from both parents, because both parents go through the meiosis process, producing sperm cells or egg cells, which then come together to form unique individuals. This is evolutionarily very significant. As soon as organisms became able to do this kind of gene shuffling and to create sperm and egg cells, you had a tremendous increase in the variation within populations. And when you have enough genetic variation, you can start to build bodies and build adaptations that you couldn't before, when everything was just cloning itself and cloning itself and cloning itself. Okay,